Hi, I'm Ayla Morehouse, and today I'll be presenting Are You My Mommy? Nest Parasitism in the Lesser Snow Goose, Anser circles. So just a quick overview. First we'll do a quick life history. What is nest parasitism, the process that we use, what our results are, conclusions and management implications, and what discoveries we've had along the way and how that's impacted our project. These first couple slides are going to be a little bit of a review. So the life history of the lesser snow goose is that their breeding grounds are in Canada and northern Alaska. They only breed in the Arctic Circle. And then they migrate down the Pacific, Central, and Western North America flyways. And a lot of the ones that we collected from all winter in Texas, right along the bay. So they are colonial nesters. They like to nest with hundreds of people around, or hundreds of other geese real close to each other. They usually have clutches of 2 to 6 eggs with one brood per season. They are precocial, so that means they come hatched, ready to go. They'll have like about 3 days worth of yolk left to feed from, and then after that they have to find food on their own. Their habitat is that they prefer ponds, shallow lakes, and coastal salt marshes, but they're expanding that region and not really sticking to that, which is leading to their overpopulation. Okay. So what is nest parasitism? Well, a parasite is the mother lays an egg outside the host mother's nest. The host mother then moves the egg into the nesting bowl. And so why are we looking at this? Why do we find it interesting? Is that we get a chance to understand evolution through something that isn't affected by natural selection. There's no sexual or fitness selection. And one of the things we're going to look into is if there's a potential relatedness to this, how they are choosing who to parasitize. So our samples were collected in the Wapausk National Park, right up in Churchill, Manitoba. The yellow dots are our actual nests, and we collected 583 shells, which we initially thought would contain maternal DNA. We eventually found that it doesn't quite hold the information that we were looking for. And then from those shells, we got 499 membranes, which holds the offspring's DNA. So what was our process? Well, first, in October of 2018, we finished doing our sample sorting, separating our shells, the maternal DNA from our membrane, the offspring DNA. And then we went on to do our prep, extracting the DNA, adding our primers, doing PCRs of the MC1R gene, and then combining everything into a little tube and sending that tube off to the UND Med building for Illumina sequencing, which was our third step. And then our fourth step was genotyping, which we just finished a couple weeks ago, which was determining the true genotype of the histogram from a histogram. And then our fifth was analyzing those by comparing offspring from the same nest against each other and looking at the genes to determine if there was a nest parasite. A lot of right now we only have offspring DNA, so we're just comparing offspring to each other. So we couldn't say this particular one is the parasite. It's just looking at the different genes, whether or not there could be one there. So what was I looking at? Well, we have these histograms. Probably can't see the numbers or anything on it, but this is the homozygous individual, so that really long bar across the top is what we're looking at. The red box is how long of a sequence that is, so it's 118 base pairs long. And then we look at this chunk down here and match it up to make sure that between our actual histogram and then the computer-generated analysis of the histogram, that we're looking at what we think is the longest and what the computer thinks is longest, making sure that checks up. Then we look at the frequency, where we had, I think this was 580, samples of the same exact sequence in this sample that were found and combined. This is important because if it's too low, we can't see if that's a true genotype, because it might be that it's just excess, it's a piece of a gene or something of that like. The long line next to it is our actual sequence, and the sequence goes much farther than is pictured. And that is important when we look at our heterozygous individuals. So we have our two longer peaks. Our first one is again at 118, and again it's at the top at the bottom. But we also have the second one at the 124 mark, and it's second also. And so then we have to look at our frequency. Is our frequency high enough? Both of these have pretty high frequencies. What would be a low frequency, at which point we would kind of dismiss that data, is if it was under 20 samples. Because again, we wouldn't be able to say for sure if that's a true genotype, if it's actually supposed to be replicating, or if it's just breakage from another piece. So how do we determine if these two are the same, or if it's just a stutter from the bigger sample? Well, we look at the sequence. And in these three boxes, and several more past what we can see, the sequences don't line up at all. They're completely different things. It's not that there's just one nucleus that, or one amino acid got deleted or changed. It's large chunks of it, so we know those are two different genes. So then, once we've got that done, we started now analyzing it by comparing it. So we had 10 primers, loci that we were looking at, and each of them, and in this example, we have 5 individuals in this nest. So then going through, I highlighted the majority of the ones that are different from the bulk of it, and we can have nests that have up to 4 different lengths because they are 2 parents, and so there could be any combination from either locus from each parent. In each of these, we only have 2, and so I would say there is no nest parasite this nest. So then we have ones like this one, where at the BCA3, all 4 of them have different genes that are being expressed. And so then at BCA9, we also have several of them that are not quite matching up, and they're really getting close to that 4 mark. But the confusing part is in that BCA9 is that we have a 106 and 106 and a 112 and a 112. And so that would be 4 if you had 2 homozygous parents. But then that 102 would throw us off on whether or not there is a parasite there. So we call this that there is a potential nest parasite here. So what does that look like? Well, if you look at it from a clutch standpoint, as you get a bigger nesting bowl, there's a higher rate of potential nest parasitism. And all of these, the salmon or pink colored, is our non-parasites, and then our blue are our parasites. The clutches with only 1 or 2 eggs in it, we said there was no parasitism because we don't have that maternal DNA yet to compare it to the mother, and so if the mother's only laying on 1 egg, it's most likely hers. And at 2, we don't have enough to compare against to say if there's a parasite or not. But if we were to take those 2, the 1 and 2 nests away, then our mean for how big our nest sizes are changes. So between 2016 and 2017, we had a surprising result of both of them having an exactly 45 to 55% ratio between your parasite and your non-parasite. And that's including our nests with 1 or 2 eggs in them. And even though there were different amounts of nests collected and different amounts of eggs collected during those 2 years, it was just very interesting. But if we took out those clutches of 1 or 2, our proportions considerably change. With 2016, we have a 71% rate of parasitism, and in 2017 there's an 86% parasitism, which is a far cry from the slide before where they are both the same rate. So what are our conclusions? Well, the larger the clutch size, the larger the proportion of parasitized nests. Parasitism changes the mean number of eggs in the clutch, and then parasitic females increase the fecundity, where the host mother has higher nest success, or has a higher potential of nest success. This is because as parasitic mothers are laying eggs into them, we could see that as the snow geese are laying up to 6 eggs, but really it could have been that they're only laying up to 4 or 5, and then the parasitic mother changed that fecundity and is adding to the population despite her physical non-commitment to the eggs. And then since there's more eggs in the nest, there's a chance that at least one of those is going to survive if there's more in the nest, even if she couldn't have laid all the eggs in it. So what are the management implications for this? Well, it changes our views on female fecundity because it's no longer just what she sits on, but also what may have been laid and the potential relatedness of those eggs that she is sitting on. The management can't rely on the idea of reducing adults also reduces egg survival, because it could have laid those eggs in other nests already before being pulled from the population. So what were some of our discoveries? Well, as I said before, our eggshells did not hold the answers we were looking for. They had maternal DNA, but they resembled the offsprings, or they resembled the membrane DNA too closely for us to say if that was truly the maternal DNA or if it was just offspring DNA or something of the like. So what we're doing now is we're going to start extracting DNA from the feather of the caerulescens. There's a blood clot there that we can extract definitive maternal DNA from, and then we can use that maternal DNA to reconfirm what we were thinking on whether or not the parasites are true parasites. We can go down to that exact individual, which one is a parasite, and then we can look at a lot of the relatedness of it. Is there a chance that this is a maybe a niece or a sister or a something along those lines, relatedness to the female sitting on the actual nest. And again, our current work is based off offspring DNA only, and so that maternal DNA would just do a lot of reconfirmation, and then a lot of the spatial awareness of is there a relation to the maternal DNA to the offsprings that are being spread across in that parasitic form. Big thank you to Dr. Brian Darby, my advisor, Sam Hervey, the graduate student that helped me a lot with this process, Dr. Susan Ellis-Valigi, for helping with the sampling and getting all that put together, and then the McNair program for helping me get into this project. Any questions? Yeah?
So these females that lay those parasitic eggs, they have their own nests also?
That's something we are curious about. We're thinking they don't. I'm sure there are some females that could lay their eggs elsewhere and then keep their own nest on top of that, and just kind of like helping spread that chance of survival of their genetics being put somewhere. But without the maternal DNA, we can't say for sure.
Yeah, because, and then how many parasitic eggs do they lay? I mean, that's all questions that we look at online.
Yep. And, yeah.
Are there other species that mirror that behavior?
It's a lot more common in precocial species rather than atricial species. And so altricial are the ones that you typically think of, like the robin's nest, like they don't have feathers or anything. In precocial species, it's a lot more common because there's that, after they're hatched, there's pretty much no parental care. And so if they're not parenting it, there's less of that impact on the females that's sitting on the nest. Because all she's going to be sitting on the nest anyways. If there's 1 or 2 extra eggs, it's not going to impact her physically as much. Any other questions?